Hi viewers, in this class we are going to discuss complement fixation apart from the antigen antibody reactions. In this topic we are going to discuss introduction to the topic, materials and reagents which are used for the complement fixation test, procedure of the complement fixation test and results, as well as interpretation of the results of the complement fixation test. Complement fixation test is used to detect the presence of either specific antibody or specific antigen in a patient's serum. It was widely used to diagnose infections, particularly with microbes that are not easily detected by culture methods. Complement fixation is still used to diagnose some viral, fungal, and rickettsial diseases. Generally, the complement fixation test is based on the competition for complement between various antigen antibody complexes and the lytic system, which consisting of red blood cell specific antibodies and red blood cells, which together with complement bring about lysis of RBC. The binding of the complement to antigen antibody complex is called as a complement fixation. This test is very sensitive technique used for the detection of antibodies in patient serum. The complement collectively refers to a group of circulatory plasma proteins present in the blood serum. In normal conditions, within the plasma and other body fluids, the complement proteins lie in an inactive state and become active after formation of antigen antibody complex. The complement fixation test is one of the major traditional tests for the demonstration of presence of specific antigens or antibodies. It requires a veritable zoo of reagents and numerous preparatory steps. There are almost as many versions as there have been users. The microtiter version developed at the Center for Disease Control and Prevention includes rigorous controls and is commonly used. The complement fixation test does not depend on hemagglutinin activity of the virus, but the antibodies must fix the complement and the sera must be free of anti-complementary activity. This is very important. The sera should not contain any anti-complementary antibodies or substances. The test has been superseded in many instances by newer tests such as enzyme immunoassay. The terminal components of the complement cascade C7, 8, 9, that is the MAC membrane attack complex, can damage cell membrane in the presence of specific antibody which fixes complement to the cell surface. In the complement fixation test, erythrocytes are used as the target cell because complement induced leakiness of the membrane can be visualized or measured calorimetrically as an increase in free hemoglobin. In the presence of specific antibodies to an infectious agent, any complement in the system is bound, leaving no residual complement for reaction with antibodies to the erythrocytes. Thus, the presence of specific antibody is indicated by the absence of hemolysis. Let us see the principle of CFT, the principle behind the complement fixation test. As we know, the complement fixation test is used to detect the presence of specific antibodies in the patient's serum. This test is based on the use of complement, a biologically labile serum factor that causes the immunocytolysis, that is, lysis of antibody coated cells. This is the principle behind the complementary fixation test. And what are the materials and reagents that are used in the CFT, complement fixation test? Here, sheep erythrocytes suspension, that is 5% suspension of washed sheep RBCs, hemolysin, rabbit anti-sheep red cell antibody, then guinea pig complement, which is free of antibodies to the agent of interest. Usually, guinea pig is the commonest source of the fresh complement. Then, barbital buffered diluents, plastic microtator plate, centrifuge adapter for the microtator plates, water bath for incubation of the plates, color standards for judging hemolysis, that is prepared by lysing various concentrations of red blood cells or red cells. These are the materials we will use in the complement fixation test. That is, simply we can say that the complement fixation test consists of an indicator system composed of predetermined amounts of the sheep red blood cells, sheep RBC, rabbit antibodies 2, which are against to sheep RBC, which is nothing but the hemolysin, and complement protein system, which is generally supplied as the guinea pig serum. The amounts of each reagent are predetermined in such a way that the introduction of complement given at a slight deficiency to SRBC or anti-SRBC complexes result in almost complete lysis of about 95% of the lysis we will achieve. During pre-incubation of that fixed amount of the complement with an antigen antibody system that is not related to SRBC and anti-SRBC, that would result in the attachment of complement to the antibody in this antigen antibody complex. This attachment or fixation of complement results in the activation of the complement cascade and the consumption of the complement components by the antigen antibody complexes takes place. Subsequent introduction of sensitized SRBC and anti-SRBC into the mixture does not result in the lysis of the SRBC because the complement has been fixed or used by the first antigen antibody system, so that it may not cause any kind of lysis. That is the main reason behind the complement fixation test and the lysis of RBCs. And procedure of the complement fixation test, it consists of two stages. The first stage involves in the complement fixation stage, and whereas the second step it is related to the indicator stage. First step related to complement fixation stage, and whereas the second step related to indicator system. What happens in the complement fixation test stage is the complement fixation test in vitro conditions, a known antigen mixed with test serum lacking complement but having antibodies. If the antibodies are specific to the antigen, formation of the immune complex occurs. Now the complement is added to the sample. The complement binds to the antigen antibody complex and is consumed in the process. To this, sheep red blood cells coated with the complement fixing antibodies are added as sensitized cells as the indicator. The lysis of indicator cells, sheep RBC, indicates the presence of the free complement in the solution due to the non-complement fixation prior to the addition of sheep RBC. The absence of specific antibodies in test serum does not lead to the formation of the immune complexes and in turn the complement fixation. As a result of complement remains in free state which lies the sheep RBC. If the antibodies are present in the test serum, they form immune complexes and thereby consume the complement by fixing it. As a result, no free complement is available to lyse the SRB. So, hemolysis indicates negative result and the non-hemolysis indicates positive result for the complement fixation. This is the procedure of the CFT. Here we are observing the complement fixation stage. A known antigen and activated patient serum are incubated with a standardized limit amount of complement. That is, patient serum is heated at 56 degrees Celsius for 30 minutes to inactivate the endogenous complement, which may disturb the test calibration. If the serum contains specific complement activating antibody, Here serum with the appropriate antibody. The complement will be activated or fixed by the antigen antibody complex. Here the complement is fixed to the antigen antibody complex. However, if there is no antibody in the patient serum, in the second step we will see here there is no antibody at all. So no complement fixation has been taken place. Okay. Here, whenever you add the sheep RBC as the indicator system and antibody, anti-sheep RBC antibody, both of them are added and thereby no hemolysis because the complement is being tied up to the antigen antibody complex. So this antigen and these antibodies are mixed together and they come in contact with the complement so that the complement is not at all in a free stage, so that there is no reaction at all, lysis at all. That indicates the presence of antibody in the patient's serum. Then coming to the here, due to the absence of the antibody, no complement fixation takes place. Now the complement is free for the attachment of the another antigen antibody complex. So here the sheep RBC and anti-sheep RBC antibody, both of them they form the antigen antibody complex, so that the complement comes in contact with the antigen antibody complex and thereby it causes the lysis of the RBC. So that indicates that no hemolysis indicates the presence of antibody in the patient's serum for a particular antigen, and hemolysis that indicates the absence of the antibody in the patient's serum against a particular antigen. Then, if you observe the results and the interpretation, so this is the diagrammatic representation. So the same thing. Results and the interpretation. So we can call it as a positive and negative. If no lysis, that is the presence of, that indicates the presence of antibody. That is fixation of complement by immune complexes. When antibody is present, it is removed from the solution. As such, the complement is available to bind the antibody on the cell in the final incubation, and thereby here there is no lysis, that indicates the presence of the antibody against a particular antigen. Whereas if hemolysis takes place, that is the lysis denotes the absence of antibody, that is negative. Lysis of the RBC found in the final incubation indicates that the absence of antibodies, lack of antibody make complement remains free. This free complement results in the lysis of the sheep RBC. So that is the results and interpretation. Simply, whenever you have the antibody against a antigen, no lysis, that shows the positive result, presence of the antibody. And whereas if there is no antibody in the patient's serum, so there is no formation of the immune complex and there is no attachment of the complement to that of the antigen antibody complex, so that it is remains free and it contact with the sheep RBC and anti-sheep RBC antibodies, then that causes the lysis. So that is due to the absence of antibody in the sample. So this is about the result and the interpretation of the CFT, complement fixation test. Advantage and disadvantages of the CFT. See the advantages. Ability to screen against a large number of viral and bacterial infections at the same time, and it is moreover economical. And see the disadvantages. It is not at all sensitive, cannot be used for immunity screening, time consuming and labor intensive, often non-specific, for example, cross reactivity between HSV and the VZV. So these are the advantage and disadvantage of the CFT. So this is all about complement fixation test, materials that are used and principle, procedure and the results and the interpretations of the results. Thank you one and all.